for another video, in this case about lipophilic hormones, right? We've been doing videos so far on the hydrophilic hormones utilizing adenylate cyclase, guanylate cyclase, or phospholipase C through G protein coupled receptors. And now we're going to look at the lipophilic hormone actions and point out the fact that they don't require a membrane-bound receptor. So we'll look at receptor locations, as well as how they tend to be tied more dramatically to transcription translation. In other words, altering the types and amounts of proteins expressed by a cell. Now, that's not to say that hydrophilic hormones can't do that too. They just do it through activation of transcription factors. This will be a little bit different, and they tend to, the lipophilic hormones tend to focus on that type of activity. All right, so let's start with our cell again. In this scenario, we don't have to have a membrane-bound receptor. And again, cells are responsive to numerous types of hormones, right? So we keep drawing, one or the other, but they probably invariably have both types of receptors. Receptors for hydrophilic hormones in the membrane, receptors for lipophilic hormones found inside the cell. And so just don't forget, and also don't forget that there's not one receptor in the cell membrane, there's thousands of them. So let's have and it doesn't matter where I draw it here. I'm going to draw my hormone molecule this time as sort of square shaped. And again, it can generally diffuse pretty readily through the lipid bilayer. So it will end up in the cytoplasm, diffusing around in the cytoplasm now. And one of the places it can run into a receptor is actually in the cytoplasm. And so we'll have a cytoplasmic receptor here. Now I do need to add two things. I'm going to add that we haven't really been too concerned about previously. I'm going to add our nucleus. So I'm going to add our DNA. It's sort of oblong here, just to sort of squash it down in the corner. And now I'm going to squash another organelle over here in this corner. And this is the one with the double-layered membrane, which would be the mitochondrion. Right, so that's the mitochondrion. This is the nucleus, right? And we've got our lipophilic hormone that, at least in one scenario, and the steroid hormones tend to do this. Well, they tend to do one of two things. They tend to bind receptors that are in the cytoplasm, or they can actually bind a receptor that's in the nucleus. And in either case, they end up, this guy would be translocated into the nucleus. I'm going to draw a little bit smaller here. Got our hormone bound to it. And what they're going to do is they're going to, this cytoplasmic hormone receptor complex will diffuse into the nucleus, or again, it could diffuse directly into the nucleus where it finds the receptor already. Again, just variation in this particular system. And that hormone receptor complex is going to bind to a segment of DNA called, this is important, the HRE. And HRE stands for hormone responsive element. Right? So in other words, it's the segment of DNA that responds to the hormone, and really more so the hormone receptor complex, but responds to it by binding to it. And that segment of DNA is really essentially like a part of the promoter sequence. In other words, it's part of the DNA that precedes a segment of DNA for the protein that we want to produce. So in other words, we have to sort of like bind and activate this promoter sequence, right, where the HRE is found, the hormone responsive element, which would activate transcription of subsequent region of DNA, giving us the protein that we want to mediate the action of this particular hormone, right? And so we would get transcription, right? In which case we would get mRNA. And again, this doesn't happen in the cytoplasm. This happens on the endoplasmic reticulum. But the mRNA gets translated into protein, right? And again, so either more of the proteins that are already existed in the cell, or different proteins that, in other words, proteins that are not currently being expressed by this particular cell. In either case, we've altered the phenotype of this cell, meaning the expression of proteins that this cell currently has. If you change the expression of the proteins, well, the proteins determine what this cell looks like and does, right? And so we've just changed the function of the cell by changing its protein expression. Okay? Now, the reason I put these, so again, largely the steroid hormones do this, but also the thyroid hormones, right? They'll also do this. But the thyroid hormones will also do, in addition to this sort of cytoplasmic or nuclear receptors and then acting through the HRE, the thyroid hormones, which I'll put over here as circles, right? They can diffuse in. Now there's actually a carrier for these as well. It's not that important, but something that actually ramps up the transport of the thyroid hormones into the cell. It just accelerates it. But they are still lipophilic and it can enter passively, but there's a carrier too. So either way, our thyroid hormones, right, so this is T3, largely T3. T3 is the active hormone. T4 is the precursor molecule, and the 3 and the 4 refer to iodide atoms that are attached to it. And so you have to chop one off of T4 to end up with the active hormone T3. So we can really follow T3 because it has much more of the actions of that group of thyroid hormones. And so the T3 hormone will enter the cell, and in this case, we're going to have it coming here. I have to draw my receptor. I'll make it blue. But in this case, right, and it's got a sort of a circular binding site here because I drew my hormone molecule as circular, right? And again, it doesn't care about these membranes either because it's lipophilic, and it binds receptors that are found in the mitochondrion. Well, that makes sense because the thyroid hormones are tied to some of the things that the mitochondria do. One of them is an increase in metabolic rate. Well, generating an increase in and tied to heat production. In other words, the more ATP we produce and use, the more heat we generate. We're very inefficient about our bond energy exchanges when we make and break ATP. So the more we make and break, the more heat energy we release. So if you get exposed to cold, we increase our thyroid hormone secretion. We increase the gene expression, right? I haven't squeezed it in here, but there's mitochondrial genes, right? And we do something very similar to what we have over here, where this hormone receptor complex would alter the expression of mitochondrial proteins, and therefore the function of the mitochondria, actually biogenesis of mitochondria, meaning we make more mitochondria. So lots of things can happen. It's tied to cell division and proliferation. It's tied to the metabolic rate through altering the ability of the mitochondrion to make ATP. There's lots of things that go in there. But let's simplify and just say it can alter metabolic function through altering the expression of mitochondrial genes through our hormone receptor complex here. Now, that's not to say that the thyroid hormones don't also act over here and do something very similar. I could put at a different HRE, I could put right, so let's have our thyroid hormone coming in here and binding a different HRE, right? Because it would probably stimulate different transcription of different proteins than the steroid hormones do, right? So I put it at a different location. So now we have pretty much the whole picture of our lipophilic hormones binding, right? Cytoplasmic, nuclear, or mitochondrial, largely for the T3, right, thyroid hormones, receptors, right, in those 1, 2, or 3 locations, and altering transcription and translation to alter the production of proteins. And again, I don't want to show this one generating proteins that end up out here because it's altering the production of proteins that are going to act in the mitochondria. So they tend to, that actions tends to stay localized here, right? But now we have the function of the lipophilic hormones diffusing into the cell, binding their receptors in one of those 3 locations, and altering the types and amounts, largely, of cellular proteins. Now, having said that, this does not mean that the lipophilic hormones can't act through signal transduction mechanisms the way we showed you for the hydrophilic hormones. They don't bind receptors out in the membrane, but they can do similar things. The rationale for that is that what I've shown you here so far about changes in transcription translation are actually pretty slow. Those actions are slow. Much slower than the signal transduction mechanisms for cyclic AMP, cyclic GMP, IP3, DAG. All of those mediated actions are pretty fast, within minutes at most. Whereas here you're talking about hours at minimum, and even more so for full-blown effects, days even, to really significantly alter the protein allocation, or not allocation, but the protein content of that cell. So what I'm getting at here is that lipophilic hormones tend to be slower acting, but they do have that ability to do fast acting things through signal transduction mechanisms, which I haven't shown here. Hydrophilics tend to be fast acting because of the tie to the signal transduction mechanisms, but they have the slower component, remember, because they can alter transcription factors, and then you're kind of in the same boat here, where you're altering transcription factors, translation, right, new proteins, and that takes hours. Okay, so we've got sort of like a fast and slow tracks for both, but hydrophilics tend to be faster acting, lipophilics tend to be slower acting overall. Okay, I think we're good. Remember, steroid hormones and thyroid hormones, those are your only 2 lipophilic classes of hormone that are going to bind these intracellular receptors. All others, all hydrophilic hormones, are going to have to bind a membrane-bound receptor with an active site that faces the extracellular space.